Hello everyone, and welcome back to the channel. Today, we're diving into one of the most contentious and pivotal moments in the history of science, the discovery of the DNA double helix. It's a story that has been told and retold, immortalized in textbooks and documentaries. When you hear DNA, the names James Watson and Francis Crick likely spring to mind, two figures who have become synonymous with this monumental breakthrough. But what if I told you that the popular version of this story is incomplete, that it glosses over a profound injustice and omits the true hero at the heart of the discovery? We're going to pull back the curtain on this scientific epic to reveal a lesser-known, yet absolutely crucial piece of the puzzle. A single ghostly image, a photograph known simply as Photo 51, and the brilliant, meticulous scientist who captured it, Rosalind Franklin. This isn't just a story about labs and test tubes, it's a gripping narrative of raw ambition, murky ethical lines, and a historical wrong that took decades to even be acknowledged. Prepare to uncover how the secret of life was revealed, and at what cost to the woman who held the key. To truly grasp the significance of this story, we have to transport ourselves back to the early 1950s. The world was still rebuilding from the ashes of World War II, but in the halls of science, a new and tense race was on. The biggest mystery of all was the very nature of life itself. Scientists knew that traits were passed down from parents to offspring. They knew about heredity, but they didn't know the mechanism. What was the molecule that carried the blueprint for a flower, a mouse, or a human being? For a long time, the best bet was on proteins, which were complex and varied. But a series of experiments, culminating in the work of Oswald Avery in 1944, had pointed to a different, stranger molecule, deoxyribonucleic acid, or DNA. The problem was nobody knew what it looked like. And in science, structure is function. To understand how DNA could possibly store and copy the vast library of genetic information, you had to know its shape. This was the grand prize, and two major teams were in the running. At the Cavendish Laboratory in Cambridge, you had the unlikely duo of James Watson, a young, brash American biologist, and Francis Crick, a brilliant British physicist. Their strength wasn't in experimentation. They were model builders, theoretical thinkers trying to solve the puzzle by fitting pieces together like a three-dimensional jigsaw. Then, about 50 miles away at King's College London, there was the experimental team. This group was tasked with the hard, hands-on work of trying to get an actual image of the DNA molecule using a powerful technique called X-ray crystallography. This is where Rosalind Franklin enters our story. She wasn't just a member of the team, she was a world-class expert in this incredibly difficult field. Recruited to King's College in 1951, she was a force of nature, precise, fiercely intelligent, and uncompromising in her scientific standards. So, how does one take a picture of a molecule? This is where Franklin's genius truly shone. X-ray crystallography is less like photography and more like deducing the shape of an object by analyzing its shadow. Imagine standing in a dark room and shining a light at an intricate crystal chandelier. You can't see the chandelier itself, but you can see the complex pattern of light and shadow it casts on the far wall. By meticulously analyzing that shadow pattern, a brilliant mind could work backward to figure out the chandelier structure. In this analogy, the DNA molecule is the chandelier, the X-rays are the light, and the photographic film captures the shadow, a pattern of spots and smudges, known as a diffraction pattern. Franklin refined this technique to an art form. She managed to prepare finer DNA fibers than anyone before her, and, crucially, discovered that DNA existed in two forms, a drier A form and a wetter form. It was her work on the B form that would produce the single most important clue in the entire race. After 100 hours of X-ray exposure, she captured the legendary Photo 51. The image was a stunningly clear, symmetrical cross of black marks. To a layperson, it meant nothing, but to a trained crystallographer, it was a revelation. It screamed helix. The data Franklin was painstakingly extracting from this and other images revealed the molecule's dimensions, the distance between the rungs of its ladder, and its fundamental helical shape. She was getting closer, methodically, piece by piece, to solving the entire structure on her own. But she was cautious, a true scientist who refused to leap to conclusions without sufficient proof. That caution, that scientific integrity, would tragically be used against her, as others, less patient and perhaps less scrupulous, were about to get a peek at her cards. This single photograph, the fruit of her labor and expertise, was about to be shown to a rival, without her knowledge and without her permission, in an act that would change the course of science history forever. The stage was set not for a collaboration, but for a heist. It is a chilling reminder that the path to discovery is not always pure, and that a single, unauthorized glance of someone else's work can be the line between forgotten genius and a Nobel Prize. What do you think is the line between scientific collaboration and intellectual theft? Let us know in the comments. Subscribe for more deep dives, and we'll see you in the next one.